Hey everyone, I'm George, and I'm a scientist at the Francis Crick Institute, and I work as a structural biologist. And this is me standing next to a Krios electron microscope, and I'll explain how I use this microscope to solve the structures of proteins. So before I explain how we solve protein structures, I'll just give you two examples of protein structure and why it's important and what it can tell us. So on the left here, I have the TRPV1 receptor. So it's a receptor found in cells in your body, and it's responsible for sensing capsaicin, which is a compound that's found in chili peppers, and that's what gives the sensation of heat when you eat chilies. So the protein is composed of subunits. It just means like different separate protein fragments, and they're shown here in green and in blue. And you can see the capsaicin molecule shown in this pink-purple color. So what I want you to take away from here is that proteins form specific shapes or folds or structures, and these structures are related to their functions. So the capsaicin molecule is recognized by fitting into this pocket on the receptor. And then on the right-hand side is the spike protein from SARS-CoV-2, which causes COVID. And these are several different structures of this protein bound to antibodies. So the spike protein is shown in this pink-purple color, and the antibodies in this orange-yellow color. Now, so by understanding the shape of the spike protein, we can understand how it performs its function, which is to allow the virus to get into cells in your body and cause disease. And we can also understand how antibodies against the virus recognize the virus or the protein, the spike protein specifically. So how exactly do we solve these structures? So there are at least three main techniques for solving protein structures. And one of these techniques, the one that I use, is cryoelectron microscopy. So obviously, as the name suggests, you need an electron microscope in order to take images of your protein sample. So this just shows you schematically what's inside that box that I was standing next to in the first slide. So you have an electron source which produces electrons. So this microscope uses electrons instead of light in order to see, because when we're looking at the structures of proteins, we're looking at they're very, very small, and light we can't produce light that has well, we can, but we can't make a microscope that uses lights that has the properties that would allow you to see these small molecules. So in this case, we have an electron source, and we have magnets that act as lenses for the electrons. So we fire the electrons onto a sample. So the sample is carried on a small grid, 3 millimeters in size. So very small. We have to pick them up with tweezers in order to manipulate them. How do we put the sample on the grid? We first purify the protein from cells. They can be mammalian cells or bacterial cells. We then apply the protein sample onto the grid and then remove excess protein using filter paper. and then we freeze the protein that's left on the grid using liquid ethane. So this liquid ethane is cooled to cryogenic temperatures. So that's around about minus 165 degrees Celsius. And the reason why we want to cool the sample or freeze the sample to this temperature is mainly because we don't want ice crystals to form, which would interfere with our imaging. And also inside the electron microscope, you have a vacuum. So if you put the sample in as a liquid sample, for example, it would just basically evaporate into the vacuum of the microscope. So we freeze it. So on this grid, we have like little holes here where the protein molecules are. When we fire the electron beam, we then form images that we can process inside the computer in order to see where the atoms are in the protein that we're looking at. And that is what the protein structure is. So essentially, the arrangement of the atoms tells us the shape of the molecule. And in short, that's what I do. So I'm looking forward to getting questions from you guys about structural biology, about cryoelectron microscopy, proteins, or about what it's like being a scientist in general. So yeah, thanks.